Hi, I'm Selena. I'm a dementia research scientist here at University College London. Come and follow me around the lab and find out what a day in the life of a scientist is like. So I'm using human stem cells to understand more about frontotemporal dementia. And the first thing I do every morning is cell culture. So I look at all my cells, make sure that they're healthy, that they're growing properly, and I feed them all. And this will probably take me all morning because there's a lot to do and I've only got one pair of hands. So these stem cells have been made from skin cells donated by patients with frontotemporal dementia. We can change stem cells into any cell type of the body, including brain cells or neurons, and try to get a better idea of what's going wrong with these neurons in the brain. So at the moment I'm just changing the media on my cells. It's quite simple really, we just take the old medium off where the cells have already used all the amino acids and other sugars and proteins that are there, and we replace it with fresh media that's been pre-warmed to body temperature so that we don't give them a heat shock. So it's 1 o'clock now, so I've finished my morning in cell culture and now I'm off to get some lunch. It's time for our weekly lab meeting now. It's my turn to present this week, so wish me luck. In lab meetings, the whole group comes together once per week and we take it in turns to each present our latest data, and then everybody gets a chance to ask questions and make suggestions on what the next steps could be. Okay, well that all seemed to go quite well, so now it's back to the lab for the afternoon. The cells that I was looking after this afternoon I'm going to analyse some of those cells, and we're performing a technique called a Western blot, which will allow us to analyse some of the proteins in those cells. So that's my gel running, and that takes about 1 hour to run, so it's time to catch up on some emails and have a cup of tea. So that's my gel finished running, and all the proteins now have been separated by size, but we need to now visualise the specific protein that we're interested in. So this shows us the results of our experiments today, and this shows us that the proteins we're looking for are there. It's the result we wanted. We're seeing it in all cell types, so we can carry on with our experiments for the rest of the week. That's me done for the day. Time to go home.